Good morning, sir.
Good morning. Good morning.
Good morning, sir.
Good morning. Swati
Yes,
is
sir.
speaking.
Swati from Hyderabad, sir.
Yeah. Very good. Very good. So I'm in the travel.
Yes, sir.
So I will leave this meeting very soon.
Okay, sir.
I have created YouTube.
Yeah.
You can continue.
Yes, sir.
And Megha ma'am is now host.
Okay, sir.
She will make Dr. Riaz as a co-host.
Okay, sir.
Okay.
Swati ma'am, I've already made you co-host.
Okay, Megha.
Okay, fine. Once sir will join, we can make him a co-host also.
Yes. Once sir joins, then I think we can give sir's introduction. Na? Yes. Then we can start the session.
Fantastic.
Good morning, Vanessa.
Good morning. Good.
Good afternoon, Riyaz sir. Welcome.
Good afternoon. Thank you.
Sir, another 3 minutes, we will start the session.
No issue.
Yes.
It's fine.
Sir, I'll give you the provision of the co-host so that you can share the screen.
Okay, fine.
Yes, yes, sir. Swati madam?
Yes, Megha.
Ma'am, another 2 minutes, we will start.
Yes.
Okay, okay, fine.
हेलो गुड मॉर्निंग फ्रेंड्स। सर मैं ऑनलाइन टीचिंग मीटिंग साइट से बात कर रही हूँ। मेरा नाम प्रिया शर्मा है सर। आपके इंटरेस्टेड है सर, आपको आपकी लोकल से हेल्प प्रोफाइल मैप प्रोवाइड किया जाएगा। आप मैम के साथ घूमना फिरना, फोन पे बातें करना, वीडियो कॉल में चैट करना, फिजिकली सेक्सुअली रिलेशनशिप रखना पसंद करते हैं सर। अगर आप इंटरेस्टेड है तो आपका नेम और एड्रेस सेंड कीजिए, आगे की प्रोसेसिंग मैं आपको बताती हूँ सर। ओके। हेलो गुड मॉर्निंग सर। सर मैं ऑनलाइन मीटिंग साइट से बात कर रही हूँ। मेरा नाम प्रिया शर्मा है सर। आपके इंटरेस्टेड है सर, आपको आपकी लोकल से हेल्प प्रोफाइल मैप प्रोवाइड किया जाएगा। आप मैप के साथ घूमना फिरना, फोन पे बातें करना, वीडियो कॉल में चैट करना, फिजिकली सेक्सुअली रिलेशनशिप रखना पसंद करते हैं सर। अगर आप इंटरेस्टेड है तो आपका नेम और एड्रेस सेंड कीजिए, आगे की प्रोसेसिंग मैं आपको बताती हूँ सर। ओके।
ಬರ್ಬೇಕು ನಾವು ಬಂದು ನಿಂತೇವೆ ಅಲ್ಲಿ, ನಾವೆಲ್ಲ ಕುಂಡಾಗದೆ.
Megha,
Yes, sir.
I'm not able to share my screen.
Sir, I have not made you the co-host yet. Just a minute, sir. Yeah. Sir, now you are the co-host. You can share your screen.
Okay, fine.
Swati madam,
Yes, Megha.
you can start the session, ma'am.
Yes. Good afternoon, everyone present here. I welcome Dr. Aravind Deshmukh, sir, President, MSCI, and our speaker, Professor Riaz Ali Zafar Ali Syed, sir, and all the participants for today's session. It's my privilege to introduce our today's speaker, Professor Riaz Ali, sir, Head Department of Biotechnology, PSGV PM's ASC College, Shahada. Sir has 25 years of teaching experience. His research areas are plant microbe interactions and microbial biopolymers. He has guided 7 PhD students and 5 currently doing under his able guidance. He has 42 paper presentations at national and international platforms. He has organized many seminars, conferences, and workshops. He has received 7 projects from UGC, DST, and Barak, worth 61 lakhs, and institutional projects worth 3 million. He has received many awards like Young Scientist Award 2008 DST, from then till Research Excellence Award 2020 Jelgaon. He stands in world's top 2% scientists as per Stanford University AD Index 2023. He is the editor-in-chief and guest editor for many renowned journals. He is a life member of Asian PGPR Society, Association of Microbiologists of India, MSI, and so on. To his credit, Sir has 276 research papers published in Scopus and Web of Science journals. He has published books with Springer, Whaley, Taylor, and Francis. Sir, we are honored to have you with us today for today's session, and this session is open to you, sir. And we welcome you for the session, sir.
Thank you, Dr. Swati. Good noon, Deshmukh sir, and all the participants. Am I audible to all?
Yes, sir, you're audible, sir.
Okay. So thank you to MBSI, Professor Deshmukh sir, and Megha and Dr. Swati for giving me this opportunity, and especially thanks to Dr. Swati for such a nice introduction, which I hardly deserve. So I'll be presenting.
Sorry, sir, we have to cut short your biodata to give it in 5 minutes. Otherwise, I have to tell many things about you. And to make a note to you, I'm not a doctorate, sir. I'm still a faculty cum research scholar. Thanks for your
Thank
input.
you.
Thank you, sir.
So I'll be presenting my work on biodegradable plastics. We call it as a polyhydroxybutyrate, PHB, or polyhydroxyalkonate. So it's a family of polyhydroxyalkonate, and there are different polymers like polyhydroxybutyrate, polyhydroxyalkonate, polyhydroxyvalerate, and so many polymers. All these polymers are produced by microorganisms. Rather to say, they are accumulated by microorganisms growing under carbon-rich growth conditions, but nitrogen deficient stress. So these polymers are in use as a biodegradable plastic, and their use can be a best solution to mitigate the plastic or polythene pollution. So let us have a look on microbial polymers versus synthetic plastic. Worldwide, around 180 million tons of synthetic plastics are produced every year. And if we consider the scenario of our country, it is around 4 million tons per year. The major countries which contribute to the production of this synthetic plastic are Europe, America, Asia, Asian continent, Australia, and India. India contribute major amount in the synthetic plastic production. It accounts highest of about 33%. So India is the third largest consumer of plastic, and the plastic industries are growing at the rate of 20% every year. The pretty thing, or you can say the thing which makes all of us worried, is the use of this plastic, and after their use, they are thrown into the environment, in the soil, in the water bodies, or you can say in the atmosphere. So that lead to the accumulation of this plastic. As you can see, if you have chance to go through or through the dumping yard roads in your city, you might have come across this scenario that the plastic bags or polythene bags are spread everywhere. So that's the very, you can say, alarming scenario. If you have visited some places like coastal areas or lakes or you can say river bodies for picnic purpose, you might have seen that the polythene bags, wrappers of various fast foods, plastic bottles, etc., they are thrown into the water bodies, and this harms the aquatic life, including the animals and plants. So these are the issues related to the use of plastic. If we use biodegradable plastic, at the place of the synthetic plastic, we can save this much number of polythene bags in our average lifetime. So it's a very big number. You can save this much number of polythene bags, and thereby you can also help in protecting the environment or avoiding the plastic pollution. So see, this is again the plastic pollution around this is the data of 2018. So it's a 5 years back data. So if you throw the plastic, this will go into the dumping yards, and then again it will decrease the soil fertility. It will affect the soil porosity. There will be less percolation of water, and it will not lead to the increase in the water table during the rainy season. So that's again a very frustrating scenario of this coastal pollution, plastic pollution particularly. If you see this lake, the entire lake is covered with plastic, different types of plastic items. And if you remember the flood of 2005 in Mumbai, the flood was not due to the other reasons, but it was due to the choke up of or blockage of gutters or drainage systems due to the accumulation of these plastic material, polythenes or plastic bottles or plastic cups or wrappers. Yeah, we all in our culture, Indian culture, we pray a cow as a mother, and what we do, we put the remaining food items or kitchen items or food material into the polythene bags, and we throw it into the environment. So these animals, they mistakenly consume the food contained in the bag along with the bag. So there is a report that if we see, or if you examine the stomach of animals in India, around 250 grams of different forms of plastic are found in their stomach. And due to this plastic accumulation in their stomach, many of the animals are also dying. So what is the solution then? How can we overcome this plastic pollution? The one suggested solution is the recycling of these polythene bags or plastics. But unfortunately, only 1% or less than 1% of these polythene bags are recycled. The reason is that recycling, it requires more economy than the production. The plastic production, if it requires, say for example, 100 rupees, then recycling may require 500 rupees to recycle this polythene. So recycling is costlier affair, and whatever the plastic produced from this recycling will also have a very less market. So it's going to be a business of loss. It's not going to be a profitable business. So Francisco was the first country to ban this plastic. Later on, all other countries have banned. Fortunately, our country has also banned the use of these polythene bags. And I guess that every municipality or every municipal corporation has also banned the use of these polythene bags. The another solution is landfilling. But if we use this plastic, synthetic plastic for landfilling, as I told you earlier, this will reduce the soil fertility as well as it will affect the soil porosity. And if the soil porosity is affected, the rainwater will not percolate down to the earth, and it will not improve the water table. And again, there will be a problem with the drought or water scarcity. So the another suggested solution is incineration. And I guess many of us in society or in colonies, we do practice this incineration. What we do, we collect the plastic bags or all other, you can say, waste, and we simply incinerate it. So, but then it's not a good practice because it leads to the emission of harmful gases like carbon dioxide, methane, and other toxic particles. the best solution to this plastic pollution is the not to use the synthetic plastic bags. And if we do this, we can also help our country in overcoming the dependency on foreign countries, particularly US, or you can say Arabian countries, and we can save around 37 million barrels of crude oil per year. This crude oil is used for making plastic. So the best solution is to use this green approach, that is use of biodegradable plastic bags, or the best way is to use cloth bags. So bioplastic are the best solution for sustainable environment. I'll be talking on polyhydroxybutyrate. My students have worked on this PhD, and 2 students have been awarded their PhD degree on this bioplastic production, characterization, and its degradation studies. So this polyhydroxybutyrate, we popularly call it as a PHB. It is accumulated by microorganisms, and it's an ideal biodegradable polymer. It is eco-friendly replacement to the harmful synthetic plastics, and it is accumulated by a wide range of bacteria which are grown under the carbon-rich environment and nitrogen stress. The 2 important things for the accumulation of PHB are the carbon-rich conditions and nitrogen stress. This biodegradable plastic is easily degraded. It is degraded by microbes present in the environment, so it can also be recycled. So it's the best solution for the plastic pollution. All microorganisms in the soil, they possesses this PHB depolymerase enzyme, and this depolymerases can degrade this polymer. And if you compare the degradation of this PHB, it is degraded within 6 months, and it is degraded to a non-harmful substances. If you compare it with the synthetic plastic, the degradation of synthetic plastic, it takes more than 500 years, and moreover, the degradation of the synthetic plastic lead to the production of other toxic byproducts. So then how to go, or how to look for whether the microbes are accumulating or they are producing this PHB or not. So there is a staining procedure, we call it as a Sudan black bee staining. So if you stain it's a lipid staining basically, as all of you know. If you stain the culture isolate with the Sudan black bee, you will find that there are you can say granules present in the cytoplasm of this bacteria. This is the electron micrograph. So here you can see all these granules are the granules of PHB. The another staining is Nile blue staining. So that staining also helps in detecting the presence of intracellular PHB molecules. And if you grow these organisms in minimal medium, which is rich in carbon but deficient in nitrogen, you will find that the organisms, they produce this polymer. And if you pour the broth after centrifugation into the Petri plate or into test tube, you will find that this type of powder can be obtained, or this type of bioplastic sheet can be obtained. So these are the you can say results of our laboratory work. The important issue in this bioplastic production is the quantity. The organisms accumulate this plastic in very less amount, and if you consider for the commercial production of this bioplastic, then this plastic has to be produced in larger amount. And for industrial production of this bioplastic, the suggested option is the cloning of this PHB producing genes into a fast growing organism like E. coli. What we did, we performed the PHB production with this alkaligenous fecalis, and we found that though it is producing PHB in more amount compared to other organisms, still the yield is not, you can say, acceptable for commercial production, as this organism is slow growing. So if we transfer these genes of alkaligenous fecalis into E. coli, it is fast growing, and in short period we can obtain more amount of PHB. So this is the result of gene cloning of PHB producing genes from alkaligenous fecalis into E. coli and its expression. So what we did, we obtained 6 fungal strains and 5 bacterial strains. After producing this PHB, we went to check whether this PHB is biodegradable or not, whether it is degraded or not. And we isolated 6 fungal cultures and 5 bacterial cultures from the soil sample, and we tested them for their ability to degrade this PHB. So we performed 2 types of assay. One was a solid environment assay, where we added PHB as the only source of carbon in the medium, and we grew these different types of organisms, fungus, Aspergillus niger, Cercospora, Aspergillus flavus, in this medium, a minimal medium, which contained PHB as the only source of carbon and nitrogen. So if you compare it with the control plate, uninoculated plate, you will find that there is a growth of these fungi, and that indicates that they were able to utilize this PHB, which was the only source of nutrient in the medium. So this is with the another fungi, Alternaria alternata, Fusarium oxysporum, Metarhizobium anisophili, and then with the alkaligenous fecalis. This was the same organism which produced PHB. The beauty of this PHB producing organism is that they also secrete PHB. Now you may ask, why, if the organism is accumulating PHB, why the same organism is degrading it? So remember this, that the organism accumulate PHB as a source of their reserve carbon and nitrogen. So under starvation condition, like in this minimal medium, when there is no nutrient available, the organism will start utilizing this PHB as a source of nutrient, and that PHB is mobilized under the influence of an enzyme that is called as PHB depolymerase. So these are the different zones of PHB solubilization with alkaligenous fecalis, with Alternaria alternata. So these were the zones of PHB solubilization. We also found Pseudomonas species, Pseudomonas aeruginosa and Pseudomonas species. They were also able to degrade this polymer, and this was a biodegradation liquid environment. So, and we also profiled the incubation period, which was required to completely degrade this PHB. And we found that this can be degraded from 3rd day of inoculation, and the degradation continues up to 15th day. So the complete degradation was possible after 15th day of inoculation. So when we compare it with the degradation in solid medium and degradation in liquid medium, we found that Aspergillus flavus has a maximum potential to degrade this polymer, and it degraded in 216 hours. And the PHB biodegradation by natural flora, when the plastic was, let me see if I have that picture or not. Yeah, the third one, what we did, we buried this plastic into the soil. So soil burial was the third degradation assay, and we found that when this pH was degraded in the when it was buried in the soil, it 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 lead to the highest degradation of PHB. It is due to the consortium, or it is due to the group of microorganisms which were present in the soil. So the degradation period was also less. Laboratory degradation, it took around 216 hours, whereas soil burial method, it took 192 hours for complete biodegradation of this PHB. Uh, Unfortunately, that slide is not in this presentation. It is somewhere in other presentation. Otherwise, I would have shown you that how this plastic sheet, PHB plastic sheet, when buried in the soil, at different stages, it led to the uh, formation of holes, then crystallization and complete degradation and disappearance of this plastic sheet from the environment. So to conclude it, the isolates which were obtained from soil, they produced around 83.7% of PHB. This is with respect to their biomass. So Pseudomonas was the organism which produced higher amount of PHB under aerobic condition. Then semi-aerobic condition, we tested different organisms and their potential and production of PHB under aerobic and semi-aerobic condition. And we found that this is the highly aerobic process, and it will produce more amount of PHB under aerobic condition than semi-aerobic or anaerobic condition. We also developed one very best method of extracting PHB from the cell biomass, and it was the 1:1 mixture of acetone and alcohol. We have published this paper in a good journal, and we are likely to patent this novel method for the best recovery of PHB. Because there are other methods we compared, and we found that the other methods, they are less efficient, or they lead to the destruction or denaturation of the structure of PHB. Whereas this acetone alcohol mixture, equal mixture of acetone or alcohol, will give more yield, as well as it will protect the chemical structure of PHB. So this method proved to be the best recovery method. The isolates which produced PHB were also able to degrade the PHB under the influence of PHB polymerase. So that's it. I have different data, but I don't think that those much details are required at this stage. Dr. Swati, I am done.
Yes, sir. Sir, now are you open for the questions, sir? Should we ask the participants?
Yes, I'll be happy to have questions.
Yes. So now this session is open for the questions. Participants, you can unmute yourself and then you can ask your question to sir.
What's the location?
Or else you can put your questions in the chat box. If not unmute, you can put your questions in the chat box. Swati ma'am, we can take up the questions from the chat box.
Yes, yes. So here we have one question, sir, from Prachi. For soil burial method, which you discussed, did you create a pit or how many grams of soil was used? What was the procedure? So this is the question from Prachi.
Yeah, I can read question of Prachi. It's a good question. Prachi, we make a pit. And in this pit, we added this bioplastic sheet, and then we covered it with the soil. And at interval of every 8 days, we observed the rate of degradation. So maybe I can share you those slides. But then it will take more time. So yes, it was the pit method we used to bury the soil, bury this plastic, and then we after every 8 days we observed this buried plastic for its degradation. Yeah, there is some question from Trupti Pandya.
Yes, sir. Can agarose waste be used as a carbon to increase PHB production?
Yes, agarose waste can be used. Agro waste can be used, and we have tested these agro waste as well as industrial waste, molasses, and other agro waste for the production of PHB using Pseudomonas, and we found that the production of these PHB from the agro waste and industrial waste is also to the extent which we produced in the minimal medium. Maximum yield of PHB was obtained in molasses, because this molasses is a rich source of carbon, and which is the prerequisite for PHB production.
Okay, sir.
Mr. Mukesh has some question.
Yeah. Yeah.
Which bioplastic is better out of cellulose based or PHB? Yes, of course, cellulose based is better.
Okay, sir. Dr. Nagarani Mahajan, yeah, you can unmute and you can ask your question.
Yeah, thank you, madam. And it was a very informative presentation, sir. And I just had few small things. Maybe I have missed it. I just wanted to know, like, what will be the production cost for these bioplastic that you have mentioned in, if we want to go it for a industrial scale of production, and what is the degradation time does it take? Like, if it is discarded, how long does it take to decompose?
A very good question. I'll answer one by one. Your first question was what is the cost of its production? Basically, we have not calculated the economics of the production. But yes, of course, this bioplastic production, if you compare it with the synthetic, with the polymer, or you can say with the polythene plastics, then it's going to be 10 times higher than the production cost of PHB. And that is the reason why this PHB is not popularized, or why it is not available at large scale in our country. But if you see, in European countries, this plastic is used for making shampoo bottles, for making toothbrush, for making moisturizer bottles. So there is a company in Europe under the name Sanara. So that Sanara company uses this type of bioplastic for their bottling purpose. Your second question was degradation period. In our study, we found that the plastic sheet, bioplastic sheet, which we buried in the soil, it was degraded. Its degradation started from 3rd day of burial, and it continued up to 15th day, and the entire degradation occurred in 3 months. And it has been reported that the full degradation or 100% degradation of this plastic can occur in water, it can occur in environment, it can occur in the under the soil or above the soil within 6 months. So 6 months is the maximum period for the complete biodegradation of this plastic.
Yeah, thank you, sir. And just like with the answer that you have given for the first question regarding the production cost, and you gave one example of a company called Sinara, which is using this technology. I just had a curiosity to know, like, if you have an idea what exactly they are doing and how they are managing the production cost, and what differently they are doing, and why we are not able to do that.
Unfortunately, I had not gone through this scenario, so I'm not be able to answer this economics.
Not regarding economics, I just wanted to know scientifically, like, what different thing they are doing. Like you said, you are trying to overexpress the gene in E. coli. So other than that, what are the strategies that we can implement so that we can speed up the process and bring that into market?
All right. The other way can be the gene cloning, as I said, or it can be the transformation of genes into plants like Arabidopsis, but it has not been yet tested. The only thing which has been tested is the gene cloning into E. coli. So that can lead
Only I
to,
had curiosity, like this much of good work that you are doing. So it should not lie in the lab itself. It should come out to the
exactly,
market. Yeah,
right.
this is where
Yeah,
like I was.
we have tested this gene cloning approach. We have cloned these genes into E. coli and we produced it at a 5-liter capacity bioreactor. So at laboratory scale bioreactors, it was okay. But then it has to be tested at industrial scale bioreactor. And that too with the agro waste like molasses. Because in laboratory condition, we are using minimal medium, which is also not a cheap option. It's a costly affair. So it has to be done with the tap water and with the molasses at industrial scale.
Okay, thank you, sir.
Thank you.
We'll take the next question, sir. This is from Trivedi. She is asking for food packaging, which material is more suitable?
Triveni, if you have some idea of National Institute of Packaging, it's a CSR laboratory, and they work on different type of packaging materials, but they are using cellulose based materials, and to less extent they are also using these bioplastics. So I can share privately, I can share you the details of this CSR laboratory. They are
Yes,
working
sir.
on different types of packaging materials, biodegradable packaging materials.
Okay, sir. Thank you.
So we have our next question from Dr. Vinay Aruskar. He is asking, how do you estimate polyhydroxybutyrate by GC and HPLC?
Dr. Vinay, there are 4 methods basically for estimating this PHB. One is spectrophotometric method, where you have to prepare a standard graph of PHB, and then you have to calculate or you have to extrapolate the amount of PHB produced by your isolate. Second one is the gravimetric method, where the PHB is extracted by acetone alcohol, and then that extract is gravimetrically measured. Third method is again with the GC, gas chromatography, where you can also check the purity and you can estimate the quantity of PHB produced. So these are the 3 popular methods which are used for estimating the amount of PHB produced by the isolates. The most convenient method is gravimetric method.
Okay, sir. He has the second question. Have you done scale-up studies in small fermenter, whether air required in fermentation?
Yeah, we have performed the you can say scale up for this PHB production, and it was restricted to 5-liter capacity bioreactor only.
Okay, sir. So we have again from Prachi, enzymes like lipases, esterases, cutenases can also degrade the bioplastics. So if I plan to work with them, how can we get such bioplastics?
Let me see her question.
Yes, sir. She's asking.
Yeah, enzymes like lipase, esterase,
Yeah.
chitinase can also degrade the bioplastic. Prachi, the best enzyme to degrade this bioplastic is PHB depolymerase. It's not the esterase, lipase, or chitinase. Because this PHB is basically a polymer, it's not chitin or it is not ester, yes, of course, it can be a lipid molecule, but then for its accurate degradation, the PHB depolymerase enzymes have been studied, and they have been reported to be the best enzymes for complete biodegradation of this plastic.
Okay, sir. We have a question from Varsha.
Prachi has some other question. I think polylactic acid she is talking about.
Yeah, like.
Yeah, that polylactic acid can be degraded by lipases or esterases. Polylactic acid is also one of the bioplastic. But if you are talking about the PHB, then it is exclusively degraded by PHB depolymerase.
Okay, sir. Thank you so much. But if I want to work with PLA plastics, sir, so how can I order them online? Because it's difficult to just find such plastics.
So you want to buy this PLA, polylactic
yes, so
acid,
that I can study the degradation with the help of an enzyme.
All right. You can get it from Sigma catalog.
Sigma catalog.
Sigma
So,
Aldrich,
okay.
yeah.
Thank you so much, sir.
Okay.
So we have our next question from Varsha. Is it easy to degrade to bioplastic or recycle it?
Recycling is easy, but if you compare it with the environmental demerits of recycling and economics of recycling, the best option will be biodegradation, because biodegradation is an eco-friendly approach. It is a cost-effective approach, whereas recycling is environmentally harmful. It will release to the either if you go for using it into the soil, you can say layering, it will affect the soil porosity, soil fertility. If you go for incineration, it will release the toxic gases. So biodegradation is always economical and environmentally friendly approach.
Okay, sir. So we have one question from Mamata. She's asking, is it possible for you to show the pictures of the pit?
Hello, Megha.
I can show the picture.
Yes, sir.
Megha is talking.
So Swati, sir.
Swati, okay. I will talk only for 2 minutes.
Yes, sir. Sure, sir.
I was out of range because I was traveling. But now I want to first thanks Dr. Riya Sayed, who is one of my good friends. He has accepted our invitation. And I know his contribution, his work is a very great. And I know him as a pioneer worker in PGPR in India. So thank you very much, Dr. Riyaz, sir, for coming on our platform. And thanks all for attending this seminar on excellent topic. Thank you. Thank you, everyone.
Thank you,
Swati.
Deshmukh sir. It's basically an honor for me.
Yes, thank you.
You are as good as my mentor.
Thank you, sir. Thank you.
Thank you, Deshmukh sir, for your
yes.
introduction.
Swati,
yes,
again, over to you.
Thank you, sir. Sir, we'll proceed with the questionnaire session, sir.
Mamta wanted to see the picture of pit.
Yes, yes.
If you allow me, I'll have to search my laptop. I can share. Or
okay.
Otherwise, I can privately share.
Maybe,
with
yes, sir.
Whatever the option you suggest.
Maybe by the end of the session, sir, once we're done
okay,
with
fine.
the questionnaire. Yes, sir. Trupti is asking again, which sample source will be effective to get the better isolates for PHB? Which sample source will be effective for better isolation of PHB?
Right. See, we isolated the PHB producing organisms from soil as well as from dumping yards of municipal dumping area of our town. And we found that the isolates which were obtained from municipal dumping yard were more efficient in accumulating PHB because that source was having was a rich source of you can say various types of diverse nutrients and diverse contaminants. So that can be the best source. Of course, soil is also a good source.
Yes, sir. And she's asking, economically at industrial level, the substrate cost is more. So how can we reduce to lower the cost?
I already spoke on this to Dr. Nagarani's question.
Yes, sir.
Molasses can be used as a best economical source for industrial scale. We have used molasses, and that too, we diluted it to 10%. And that dilution was not done with the distilled water or deionized water. It was done with the tap water. So again, if you are using tap water, it's going to be cost effective. If you are using molasses, it's also cost effective. And if you are diluting it 10 times, then it is more cost effective. So 10 times molasses, if diluted to 10 times, it will be more cost effective.
Yes, sir. So we have our next question from Chandru. Bioplastic is completely from the microorganisms, or it is to be added with additives like plasticizers, like PET plastics, etc.
No, addition of PET plastics will interfere with the biodegradation of bioplastic. So only bioplastic, if used, it is completely biodegradable. If we use PET plastics, then it will interfere with the biodegradation of this PHP.
Okay. So we have a question from Shilpa, Shilpi Prabha. We use bioplastic for packing warm food, will not melt it into and enter into our body and do harm anyhow to humans or if eaten by animals by mistake. Will it melt?
Can you please come again for this question?
Yes, sir. She's asking, will the warm food melt the bioplastic what we prepare and will it be harmful for humans or animals?
Very good question, Shilpi. This plastic is highly thermostable. It can resist temperature of about 200 degrees centigrade. I have not shared with the physicochemical properties. I have not shared the physicochemical properties of this plastic, but it is thermostable. It can withstand the temperature of around about 200 degrees centigrade. That's the one thing. It's not going to harm the human or animals as it is completely biodegradable.
Okay, sir. So we have again from Triveni. She's asking, is it home compostable or industrial composting?
It is home compostable also.
Okay. Yes, sir. So we are done with the questions from the chat box. Sir, I have questions from my side.
Yes, please.
Yeah. So when we review with the papers, we see that in the conclusion part or in the future prospects, to work more on strengthening this bioplastic because it's not having that much strength to drag out into a film or a bottle. So how can we go about with that, sir? How can we strengthen our bioplastic, what we generate at the lab front?
I'm sorry, Swati, can you please come again for this question?
Yes, sir. The bioplastic, whatever we produce at the lab, whether it's in the form of a film or a powder, how can
whether it is what?
Form of a thin film or a powder of bioplastic PHB, how can we strengthen it so that we can have a firm plastic?
See, you can obtain it in the crystallized form as well, and you can get its a sheet.
Okay.
So once the incubation period is over, and you have centrifuged the broth, separated the biomass, this supernatant you can spread either into the tray or into Petri plate. And as I have shown you in previous slide, maybe where it is. Yeah, can you see this one?
Yes, sir.
So this is the crystal form, and these are the sheet forms. But
Yeah.
to strengthen it, you need to add it with the some what we call composites,
Yeah.
like cellulose composites and other things. So its strength will be increased.
Okay. So still, when we have produced this bioplastic at the lab, somehow we are not able to do it at the industrial level, no, sir?
Yeah,
There are many limitations because of
right.
its strength. And the films what we generate are also very fragile.
Right. Yeah, this is the slide someone was asking.
Yeah.
You see, this is the picture of not exactly the pit is visible, but you can see the soil, and this is the PHB which we obtained from Alkaligenous fecalis. We spread the supernatant into Petri plate and we found this shape of plastic. So initially, this was the structure of plastic from plate. Then after burying it into the soil, it resulted into the cracks and formation of holes. And then this was the structure after 15 days. Unfortunately, I don't have the other structure where we obtained a complete biodegradation of this plastic. So these are the few stages of plastic degradation. This is the original. This one is partly, or you can say, beginning of biodegradation. And this is the good extent of biodegradation.
Yes, sir. Triveni is asking by which method. She's asking by which method have bioplastic film is prepared, casting or extrusion?
Casting is the simple method. It is prepared by simply pouring into the plate or into the tray. So if you pour into the plate, you will get this type of structure. If you pour into the tray, you will get a rectangular sheet. So it's a simply casting. It's simply pouring of the broth.
Yeah. Trupti is asking, sir, how can this sheet be formed, as I have always got it as a powder form only?
I don't know what issues, what might be the problem you have faced. But when we tried and when we extracted, see, if you are going to extract it with the acetone alcohol, you are going to get where it is this type of powder. So after extraction, you will get this type of powder. But if you simply pour the supernatant, the broth from where you have separated the biomass, and then remaining this supernatant, if you pour into Petri plate, you are likely to get this type of structure. So don't go for acetone alcohol extraction. Simply pour the supernatant into the plate and you will get this type of sheet. Of course, this sheet will have some remnants of media. You can see some components of media are adhered to this plastic sheet.
So this is molasses minimal media, sir?
No, it's salt minimal media, not molasses.
Okay, salt minimal medium.
Minimal medium containing salt only,
Yeah.
and high amount of glucose or carbohydrate, and completely deprived of nitrogen. No nitrogen source.
Okay. And then your organism.
Yeah, then you have to inoculate it.
Yeah, inoculate
The
with the
log
organism.
phase culture of
Yes,
organism.
sir. Yes, sir. So it was for 24 hours incubation, sir?
It was for 36 hours, after 36 hours.
36 hours of
Yeah.
incubation. Then simply centrifugation.
Yeah, centrifuge the broth, separate out the biomass, and then supernatant you can pour into the plate or into the tube, and you can get this type of structure. If in tube or in plate, this can be the structure.
Yes, sir. So how much time it will take for that solidification, sir?
It takes around 1 or 2 hours.
Okay. Yeah.
Because the rest of the liquid, rest of the material will simply separate out as a, you can say, water.
Okay. This is something simple that what we have referred
Yeah,
in the research.
but then you have to keep
Yeah.
it at under the oven at around 40 degrees centigrade if you want to make faster evaporation.
Okay. So in the oven, we have to place it at 40 degrees centigrade.
Right. Not above 40 degrees centigrade.
Yeah. We should definitely try this out, sir.
Yeah.
Any more questions from the participants?
Swati ma'am,
Yes, Megha.
we can take the questions for another 5 to 10 minutes because it may not be possible even for sir to answer each and every.
Yes.
So I guess the participants can post the questions in the PhD Research Scholar group, and whatever possible we will do for the same.
Yes.
And sir, it's a request, if you can share your PPT for to us, we can provide it to all the participants. It will be very helpful.
This PPT, can you download from this meeting, or I have to share it to your
No, sir, we
WhatsApp
can't
group?
download. It will be better if you can share us through mail or through WhatsApp.
Okay, I will share to your WhatsApp.
Okay, sir, sure.
Okay.
So
I would suggest the participants to reach out me for any sort of query related to this PhD or anything related to publications. Because to be very frank, I am very good in publishing. Last year, I published 54 papers. So,
that's so
an average
great,
of
sir.
average of 1 paper per week.
Oh my God.
That too in a good impact factor journals, above 5 impact factor.
Yes, sir.
And this is possible because I have a very good collaboration with the foreign scientists, foreign researchers. So this helps me in producing papers, and I wish that all the participants or my Indian fellows should take the advantage of this thing.
Sure, sir. Sure. Thank you so much, sir, for your valuable time. And thank you for accepting our invitation and being with us today for this session. I hope all the participants have received good inputs from you. And thank you for the extension of your help for the further queries regarding the paper publication and the work related to PhD. Thank you so much, sir. I hope all the participants have benefited from the presentation and we'll look forward for the future endeavors. Megha, over to you, Megha.
Yes, ma'am. Sir, it was really a great opportunity to all of us to listen to you. And we are very thankful from the entire Research Scholar Society as well as the MBSI. Thanks a lot, sir. And it will be very
Thanks
helpful.
to MBSI, thanks to Mega for reaching out to me. My email ID is given here. And
Sure, sir.
my WhatsApp number I will also share in your scholars group. It's also there in the MBSI group. So you can reach, all participants can reach me anytime for any sort of help. Thanks to all.
Thank you so much, sir.
Thank you, sir. Thank you once again.
Bye-bye.
Thank you, Swati.
Thank you, Megha. Thank you, sir. Thank you for your patient answering for all the questions. Yeah.
Bye bye. Thank you.
Yeah. Thank you,
Thank
sir.
Thank you to all the participants who joined the session.